Hello, everyone. Welcome. My name is Christopher, and today I'll be talking to you about my project, which is on genome interval intersection. So to actually begin, I'm going to sort of like double back on what exactly a genome is. So say, for example, a human genome holds all the human genetic information, which means that we know exactly where genes are, we know exactly where the non-coding regions are, we know exactly where what genes are expressing where and what not. And a genome interval really is really the chromosome position and the start and end coordinates. Say, for example, we have a gene, and then we know the where which chromosome that gene lies in, and then the start and end coordinate of that gene. And a genome interval intersection really is where one gene intersects another gene. Say, for example, we have a gene that goes from 10 to 100, and we have another gene that goes from 15 to 80. In that case, that will be a sort of like a complete intersection. Or we have another gene that goes from 10 to 100, or another one that goes from 50 to 200. In that case, that would be a partial intersection. So my goal really is to find these genome intervals using applying this NLP method, because the way this has been done is by using algorithms. So these algorithms really mark the start coordinate and then mark the end coordinate, and then sort of like sometimes you might have to sort initially to be able to do this, and then you have to go through everything. But what I'm sort of like trying to do is by using NLP method, I'm hoping that it would sort of like improve the amount of intersections that can be found in one computation. So an important part of sort of like the finding genome interval intersections is that it can help with sort of like, say you have a variant in a gene, which means that, and this variant is causing you to have cancer or something. By using genome interval intersection, we can detect if there are any differences between your particular gene and someone else's gene. Um, it's used in precision medicine to know what, like population genetics to know, oh, has there been anyone in the population who has this kind of disease? And by using that, we're able to sort of like find your best fit treatment in terms of in terms of that particular disease. So my goal really is to use another method in finding this sort of like genome interval intersection that um, isn't what has been done before. So this is what the data looks like. This is a sample of what the data looks like itself. So we have the chromosome position, the start coordinate, the end coordinate, and then the gene, and then the strand. So I'll explain. So this data is from the Human Reference Genome. So the Human Reference Genome was a project done, started in the 90s and finished sometime last year, and it really holds every genetic information of humans. So say, for example, we have this gene OR4F5. We know that this gene is in chromosome 1. We know that this is the start coordinate, and we know that this is its end coordinate. And we have that for every gene and every non-coding region and all of that. And then this strand really talks about, this is a little bit too biological and is not necessary for this, but it's really just talked about if it's sort of like going in an upward direction or a downward direction, but this is not necessary for the computation that I'm about. So the only information I really need for this interval intersection is the chromosomal position, the start coordinate, and then the end coordinate. And that is sort of like what I did here, is really just to trim off everything that I don't need, thereby leaving everything that I need. But I do think that maybe as time goes on, like this project is still ongoing, and maybe as time goes on, I'm going to have the gene name in there, because when an intersection occurs, I want it to pull out the gene name directly instead of me doing that manually. So this is the original sample of what the data look like. And then this is a sample of what I need for the project. So I started first by tokenizing the data, and then we see here the sample of the tokenized data. Um, We see here the start coordinate. We see here the chromosomal position, and then we see the start coordinate and the end coordinate. And we have that for all the genes available in this reference genome. And then the next thing I did was sort of like from the tokenized data, create um, and also I would like to point out the reason why I tokenized this because most of the vocabulary I created, most of them were already integers. And then the chromosomal position itself are integers, but there are two that aren't, which is the X and Y chromosome. And I needed that to also be an indices format. So I had to sort of like create a chromosome, create tokenized data and tokenized vocabulary for everything to be able to achieve that. So, and the next step of what I did was I used this data to sort of like create embeddings. So we see for the chromosomal position, the start coordinate, chromosomal position, and the end coordinate, we see sort of like it's embedding here. And what I try to achieve with this embedding is that sort of learn, um, represent the tokenized data, and then represent how this tokenized data is sort of like relating to other data. Because the goal of what I'm going to use really is instead of really outrightly finding intersection between one gene or another gene or sort of, I sort of like find similarity really, how similar are two genes to each other. And then if we go down a little bit, we see an example of what the output is. Of course, I'm still going to explain how this was achieved, but just here we see sort of like this sample example of what this is, where my resulting embedding really is for every gene, we find another gene that intersects with it. Every gene, we find another gene that intersects. So I use two models. One of them is really a Bayes model, which is really just a sequential neural network. And another one is a CMS model. The Bayes model, I wanted it in a way to capture because in my Bayes model, I included the embedding. So I wanted it to sort of like capture and be able to retain the information that I'm trying to embed. And then I use the CMS model, which is really for every two entities or genes or whatever that it encounters, it determines if they are related or not, how similar they are or not. And I think I had a thresh of like 0.6 or a 0.7. I don't think I included that here, but I have a threshold for how similar a gene would be to another gene. Um, and I actually sort of like this test some of these out, and some of them are quite accurate. So at any given time, the CMS model takes two genes, which I like positive genes that sort of like are similar to each other, and it's caused them a one. And also, it's not just training to notice, sort of like training to know when two genes are related to each other. It's also training to know when two genes are not related to each other. So at the same time, not only is it sort of checking the similarities between this gene, it is also checking the similarity between these genes, and it's able to be update and train. So here is sort of like sample of the base model, and here is the summary of the output model. So for my results, I trained them for 50 epochs, and we see here that the loss actually for both the positive pairs and the negative pairs, because it's training both of them simultaneously, we see here that the loss actually does go down as the epoch sort of like progresses. Um, but then something um, interesting happens when I calculated the accuracy. I found out that the accuracy doesn't go beyond 31%, which is troubling. Um, I think it might be, I'm just sort of like, I think it might be a case of maybe the Siamese model might not be the best suited fit for this, or maybe I need to do something else intermediately to get to this point. As this is sort of like novel approach to this problem. Can't reference anything to know what is going on wrong or what is going right. I can only sort of like troubleshoot them as I go on. And of course, this sort of like will build into a bigger project. And I will try to improve on this and see what I can change as time goes on. So we see here that it just really just calculates most at 1% at any given time. So to conclude, this project was sort of genome interval intersection, and it's sort of instead of using algorithms to find this intersection, I'm using NLP approaches by tokenizing, embedding, and finding the similarity and dissimilarity between these embeddings. And then I was able to achieve some results, but not sort of the high level or as much as I would like. And this project, I would continue to sort of like improve as time goes on. So thank you so much.